esteemed colleague, we are delighted to have you here. Welcome, Professor Huber.
Good morning. Do you understand me in the back? It's a pleasure to be here to see yesterday the MIPT and the CAMRAR and look forward to in the coming years to see how it develops their brave, keen plans they have. Well, the motto is, as I read, breakthroughs of the 21st century. Now, this, I think our grandchildren can charge at the end of the century simply because it is the essence of discoveries that they are not predictable. And they come like lightning out of the blue sky. So we will see what our grandchildren say about the breakthroughs. But I can tell you about the breakthroughs in the 20th century, and this is what I would like to do with the first few slides. The breakthroughs in my own field of research, which is structural biology and crystallography. And let me begin with the beginning. What you see here is the first X-ray photograph of a crystal. It's really an ugly picture, a diffuse diffraction pattern, diffuse spots, but it meant a revolution in, it founded crystallography. It meant a revolution in chemistry because in the development we were able to see atoms, to see molecules, and it revolutionized biology because we also were then later able to see the large biomacromolecules at atomic resolution. Now this is was an experiment that Max von Laue, a physicist, did in Munich at the university. His publication in June 1912, a German text printed in the Berichte der Bayerischen Akademie der Wissenschaften, a journal nobody reads. So it has an impact factor 0 in modern terminology. Yet it led to a Nobel Prize 2 years later. So it was clearly, very quickly recognized as a breakthrough. This is Laue and Count Bernadotte at one of these wonderful Lindau meetings of Nobel laureates. I just can recommend to the students and their mentors to try to find a place there. It is organized every year, again in beginning of July this year, so try to go there. I try to be there regularly. I meet large delegations from foreign countries of students. There are about 500 to 700 students from all over the world, delegations from the US, from China, from India, even very small countries like Mauritius, this tiny island in the Indian Ocean, sent 2 students. But I miss the Russians. I really do miss them. So try to send your students there. It's a unique experience for them. They meet 30, 35 Nobel laureates in a wonderful place of my home country, Bavaria, island on a lake close to the Alps. So they will enjoy. Now, These are the founders of X-ray diffraction. There is Röntgen, who discovered the X-rays in Würzburg. Fantastic story about his experiment because he carried out an experiment many physicists were doing around the world, but they did not see the effect. Röntgen had a sharp vision, and he saw it and interpreted it correctly. This is Laue. So the rumor about Laue's experiment went to England, where father and son Bragg worked. They were chemists, and they immediately grasped the importance of Laue's discovery, offering a possibility to determine crystal structures, very simple ones first. But this was the first experiment to see atoms, so revolutionizing chemistry. And this is Max Perutz, also working in Cambridge, England. The Braggs founded a school where then revolutionary discoveries were made in Cambridge. And the father of protein crystallography is Max Perutz, who was able to decipher these complex diffraction patterns. Now, biological crystallography. So, of course, first one was just able to interpret the simple diffraction patterns of simple molecular of simple crystal structures, molecular crystal structures. But then with the discovery of Max Perutz, then one was able to analyze the diffraction patterns of large proteins. Now what you see here is the relatively slow growth of biological crystallography. It was just a handful of protein structures determined in the late 60s. I started protein crystallography 68 around, was a handful of such structures. Slow growth until about 1990, and then you see the exponential, the explosive growth. So that has a reason. The reason is the technological advances. The reason is another reason is that one understood that biology needs to see the molecules at the atomic level, and then lately, and I will speak on that in the last paragraph of my presentation, the application in medicine and biotechnology, drug design, using protein structures. So what were the major technological advances? We had now recombinant proteins available for rare proteins. We have crystallization robots. We can work with tiny amounts of protein and use robots for an automatic setup of crystallization trials. We make thousands, 10,000s of such crystallization trials in plates automatically, and then watch these plates after a week or 2 weeks to see whether we can get crystals. We then go back. If there is no crystalline material appearing, then we go back and modify the proteins, cut off N and C termini and make other changes that do not affect the function, but they do affect the crystallization properties. Now we do have the very powerful X-ray sources. This is Röntgen's X-ray source, a primitive machine, a high voltage generator that you see here, discharge tube, a vacuum pump. By the way, this original instrument, which revolutionized not only chemistry, physics, technology, the X-rays, what would we do without X-rays, you can see on display in Munich at the Deutsche Museum, which is actually the mother of all technical museums in the world. Now we use synchrotrons that the physicists built for their purposes, particle physics. And then it was found out that during the course of the electrons, these large rings, powerful X-rays are generated. So this is an experiment as we do it now. We rotate the crystal in front of the X-ray beam and record snapshots that take fractions of a second to make, evaluate the intensities of the location and intensities of these spots such that after a few minutes we do have a complete data set. So our data set are the, we go back, are the intensities and location of these spots. We usually have millions of them in one run. Now there was a problem. So the proteins worked on until, let's say, 1990 were all soluble proteins, water soluble. And, but a large proportion of the proteins are embedded in membranes. And they were thought, first, difficult to prepare, and second, non-crystallizable. Now, for reasons I have no time to discuss, we worked with a membrane protein which is called the photosynthetic reaction center. It's the biological photocell in purple bacteria, and we're able to crystallize and obtain a beautiful high-resolution diffraction pattern, analyze the crystal structure, which at that time, when we published 1985, was the largest asymmetric protein analyzed. So it consists of 4 components. These middle components, light blue and blue, are embedded in the membrane. And these are the chlorophylls. So this is the primary machine which converts sunlight into an electrical current. So the primary step in photosynthesis. Fascinating also because it is the fastest chemical reaction you can think of. Electrons are transferred in picoseconds from the donor, which is here, to the acceptor, which is down here. There was a slow progress from our publication 85, Nobel Prize 88. Again, some delay period of at least 10 years until the next membrane protein structures were determined. But now we read about fascinating membrane proteins almost every second week in the top journals. Nobel Prizes have been given. The last one, 2012, Lefkowitz and Kobilka for G-protein coupled receptor. These are particularly important structures because they represent a large proportion of the drug targets. So we would like to see how they look like to help in drug development. There are other techniques in structural biology, X-ray diffraction, a fascinating development in electron microscopy we face now and again. If you follow literature, you see almost every week a new near atomic resolution EM structure of large aggregates. Again, the reason for that development is technological advances, new detectors, new electron guns, faster computers. So also the combination of X-ray analysis of fragments of these large aggregates at high resolution, and then the fitting of these atomically resolved models into the shape given by electron microscopy is a fascinating development. There is NMR, no time to discuss about this. This illustrates an example of our work, very recent example, how we do it. So we get the shape by EM. We had atomic structure of fragments of this, which we then could fit into the shape to get a high-resolution structure by a combination of electron microscopy and X-ray crystallography. There is small X-ray scattering. Of course, for crystallography, we need crystals. But there is X-ray diffraction also from biomolecules in solution, which do not give atomic resolution, but they give us information on the shape. This is one of the experiments we had done a year or two ago to discriminate between various models of actin bound to a nucleator molecule. Again, no time to discuss it, but there is great development also in solution X-ray scattering. Now I come to the structure-based ligand design, so the focus of my presentation, shown with the example of thrombin, which is the major clotting enzyme and a major focus of drug design in cardiovascular diseases. So this is the APO enzyme, this is the ligand binding site, and we then, by simply looking at it, can design a molecule, a small molecule that might fit tightly and then prevent the attachment of a substrate, inhibit the enzyme. This is an experiment we had done. But instead of just looking at it in the graphics system, we can use programs that are now available which scan the whole protein surface for possible docking sites. This is such a program that is available. It analyzes depressions, cavities in the protein according to its electrostatic properties, its stereochemical properties, hydrogen bonding, what have you. And then by virtual screening of large chemical libraries, it suggests a possible ligand. Now, we shouldn't believe that. Well, we should work with it, but try to confirm. So this is where the chemists come in eventually, and assays in order to verify what the program has suggested. This is an interesting slide that shows how large the chemical space is. So how large these chemical libraries are, 10 to the 16, incredibly large number compared to the protein space. So we can screen for a particular protein virtually these large libraries and pick out those molecules that might bind to a target protein. Of course, again, this has to be verified by assays at least, but also by structural biology. There is cross-reactivity, no time to discuss it. So let me pick out and illustrate what we are doing in various fields, and begin with agroscience. I was involved in that in work in that field together with Bayer crop science company since many years. So crop science would like to develop herbicides, fungicides, and insecticides. And they have now adopted the instruments and the methods that one uses for pharma research for high throughput screening of very large libraries, millions in a short time on intact plants that you can see. Now, now we joined these efforts with a number of systems, and I show one a collaboration with a Bayer company on a most prominent herbicide. The herbicide targets this enzyme, protoporphyrinogen oxidase, protoporphyrinogen 9 oxidase, which is a key enzyme in the pathway to heme and chlorophyll. Now what happens if PPO does not function properly first or has been inhibited? Now there are patients that have non-functional PPO, and they develop this very substantial photosensitivity in their skin. Now if one sprays PPO inhibitors onto plants, they develop symptoms which are very similar to what one sees in this human skin, increased photosensitivity, which then leads to the death of these wheat plants. So we looked at PPO. It's a dimeric molecule. This is the active site where protoporphyrinogen 9 is oxidized with the help of flavin. This is a single subunit where you see the cofactors. It is in the membrane associated with ferrochelatase, such that the product made by PPO, which is protoporphyrin, is internally transferred into this ferrochelatase, where it is conjugated with iron and heme is released. So an assembly line that we see here in biology. Now, what can we do? Of course, we crystallize the enzyme with a prominent herbicide, which you see here. You see here the enzyme, just the active site. Blue is the substrate, protoporphyrinogen. Green is the herbicide. So it's quite clear that there is competitive binding, competitive inhibition. Now, this structure became important with the discovery that in the US, a herbicide-resistant Amaranthus tuberculatus mutant has been found, which causes great damage to crops in the US. And the reason for this herbicide resistance was mapped to the deletion of a single residue, glycine 178. This is the model, and we see that it is very close to the herbicide binding site. It causes a distortion of the structure such that we can understand why herbicide is no longer bound. So this would in principle offer a way to make modifications of the herbicide to overcome this resistance in a structure-based approach. Pharma research, I was involved since a long time in kinases, which are, as you will see later, the major protein class in cancer research and cancer drug development. Now this is a scheme that I don't have time to explain, but in cancers there is often a disruption of cell signaling, and this is due to malfunction of kinases. So the approach is there to identify for a certain cancer the kinase that plays a major role, and then develop inhibitors against it. Now the problem with kinases and drug design is that there are 500 of them in the human genome, and they all look very similar. But we need specificity, and this is the major problem. So how can we approach that? Again, by using protein structure and crystallography. As I said, we had been involved in a number of such kinase drug design campaigns. And the last one, just a few years ago, was cyclin CDK8 cyclin C, which is a transcriptional kinase, and it is known as a potent oncogen. So this is the structure that we determined of this, the kinase, that is the cyclin. So the structure explained why this kinase is quite specifically binding to cyclin C. It also shows an inhibitor bound, which we then set out to develop further with the aim less to improve affinity, but more improving residence time. That is elongation, increase of residence time. And so we set out the program by making, synthesizing and assaying and structure determination of a anchor compound and elongating a side chain and checking affinity and residence time. Many hundreds of compounds have been made. The most promising one studied by crystallography to find out what determines the residence time, and it turns out that it is the so-called front pocket binding that is decisive. But by this campaign we were able to increase the residence time from less than a minute to 1,600 minutes. Again, structure-driven. Blood coagulation, also a field I was involved from the beginning of my research, starting actually with the basic pancreatic trypsin inhibitor. Michael Levitt mentioned it as a model for the first molecular dynamic simulation. So we had determined in these late 60s the first structure of that and kept this field and the interest in this field of proteases and natural protease inhibitors. So I just pick out one, that the blood coagulation cascade thrombin inhibitor development. So first hit, not already a relatively specific binder, but not perfect because this residue does not fill its pocket. So if we enlarge it from a phenyl to a naphthyl group, then we gain a factor of 100. Though this kind of approach has been used for the design and the development of a novel factor 10A inhibitor by Bayer, Xarelto, which is already a blockbuster in the clinic. This is factor 10A that we had determined many years ago. It has the enzyme component, it has spacer parts, and a membrane anchor. This is where the inhibitor binds. We can look at what nature does. Now, the blood-sucking animals, leech, are full of coagulation inhibitors, because if they suck themselves full of blood and the blood would coagulate, they would solidify, and they cannot digest the ingested blood. So they inhibit blood coagulation very effectively, of course, using the means that nature has, namely small proteins. This is hirudin. a small protein that very tightly, very specifically binds to thrombin. Now this is in fact an approved anticoagulant drug, Hirodin itself, but also variants, semi-synthetic variants that use the C-terminus and a small chemical anchor group. These are others in this bug and in the tick. So development to the clinic is, I think, happened only with the Hirodin. Now I come closer to the translation. This nice cartoon shows where the problem is. There is basic academic research. They have the ideas. They have the people. And they shout, now, look at us. We have a wonderful idea. There is industry. They say, well, it's not developed enough, so go further, and then we will license it and we'll develop it. So there is this gap, and how to bridge this gap, which is called, has to do with financing. It has to do with the lack of innovation on the industry side, and the difficulties with translation from basic research to industry. This is where universities should help. I think MIPT and ChemRAR are working in this direction. Now my own institution, which is the Max Planck Society, set up in Dortmund a lead discovery center which tries to bridge this gap. So developing ideas from basic research, mostly from Max Planck Institutes, to a stage where it becomes interesting for industry. Now I was involved in that, am still involved in that, by suggesting to the LDC a particular target on which we had worked since 10 years already, namely the huge intracellular proteases, which is called the proteasome. It is a molecule that is involved in a very large number and of physiological processes, essential physiological processes. For instance, like the executioner of the ubiquitin pathway. You know that the discovery of ubiquitination has been honored with a Nobel Prize in 2004. Now what happens here is that proteins that are to be degraded, for instance, cell cycle signaling molecules, must be removed very quickly from these cells in order for the cell cycle to progress. And the labeling of these molecules is by this polyubiquitin chain, and the degradation of these proteins is by the proteasome. That was the structure that we worked on, first publishing in 15 years ago. It's a huge protein. I should add that not only is the proteasome involved in this ubiquitin conjugation pathway, but it's also the waste cleaner. So it recognizes denatured proteins and degrades them into peptides, which, when coming from an infected cell, a virally infected cell, then these peptides are antigenic and they are transferred to the cell surface and trigger the immune response. There is no T-cell immune response without proteasome. So a very essential molecule. This was the structure that we had first determined in 95, and then 97, following then by mammalian proteasomes some year later. It's a beautiful example of evolution. Archaeal, bacterial, and mammalian proteasomes have the same architecture, you can see. So consisting of 28 subunits. But the chemistry of the primitive version is much simpler. There are only 2 different subunits, 7 alpha, 7 beta, 7 beta, 7 alpha, all identical. Now, in mammalian yeast, all these alphas and all the betas are different. So this is how evolution works. It generates specificity, but keeps the main structure and mechanism. There are many examples of that seen by structural biology. Well, now, the proteasome and as a drug target. This was a big surprise because of the essential role of the proteasome. As I told you, it is involved in essential cellular processes. How can it be a drug target? Now, it was found serendipity by a small American company who found that this simple boronate, peptide, and boronic acid, which, as we saw by crystallography, binds covalently to the N-terminal threonine. I had no time to tell you that the essential residue in enzymatically active residue in these proteins is an N-terminal threonine. So it isolates the N-terminal threonine and inhibits. And it was found to offer a new strategy against cancer, particularly against multiple myeloma. Big business. We had no share in that. We just presented the basic structures. But a consequence of this commercial success was that people all around the world jumped on this new target and screened their libraries. Hundreds of thousands of compounds have been screened, chemically synthesized compounds, and in particular natural compound libraries. Many of these had been sent to us to find out how they bind. This is a very small collection, but what you see in yellow here, these are natural compounds from natural compound libraries, which are very potent proteasome inhibitors. You see how they all do have a head group which targets the N-terminal threonine, a lactone, an epoxyketone, an aldehyde, an unsaturated amide bond. So, big surprise and enormous activity in the pharma field. This just shows a collaboration with plant biologists. I'm happy to see Klaus Palme here. So we collaborated with his colleague in Zurich, Robert Dudler, who had found that a plant pathogen which kills bean plants requires a virulence factor, which is this molecule, and we found that it inhibits the plant proteasome that can be shown by crystallography, and it can be shown by functional data because of the accumulation of cyclin B1 polyubiquitin conjugates. So this again is now, has been synthesized and modified and so on and so forth. Well, back to the lead discovery center of the Max Planck Society. Now, we did not want to interfere with the worldwide work on constitutive proteasome inhibitors to compete with big pharma that a small academic group cannot do. So we focused on a proteasome variant which is called the immune proteasome. have no time to discuss that, but this is a variant of the proteasome which is almost identical to the constitutive proteasome, but has slightly different functional properties, and it is expressed in hematopoietic cells. So what we are now engaged in is to develop immune proteasome-specific inhibitors, which do not touch the constitutive proteasome. Now, why is this important? It is important because it turned out that inhibition of the constitutive proteasome is, in principle, a new strategy to fight cancer, but it is extremely toxic. While it is already shown that immune-specific, proteasome-specific inhibitors are not toxic, and they target other diseases, namely autoimmune diseases. So this is what we are continuing now, and we fortunately have progressed so far that Big Pharma has become interested and joined the operation. Right. Do I have 5 minutes more? Then tell you about my engagement in foundation of biotech. And the first one I co-founded 15 years ago, which is now 80 co-workers biotech company, which offers services for Big Pharma. And the services they offer are protein production assays and structure analysis, and sell this to Big Pharma. So Big Pharma comes, they discover the new target, or an old target, but a new compound, and in order to develop that further, they need structure information to give chemists an advice. So for that, they need structure information, as I said. Now, Big Pharma usually does have structural biology groups in-house, but there is already since several years a strong tendency to reduce these substantially and to buy these services. So that is water on the mill of these specialized biotech companies like Proteus. So they did not need actually an investor because they live on their income by selling the information. Now this is the structures, so-called gallery structures, that Proteus offers. So these are targets that have been completely defined by them. They have the protein, they have crystals, they have crystal structures. So this is ready to go. If somebody comes and would like to know how his compound binds, then this information can be delivered in 2 or 3 weeks because everything is ready to go. Now what you see here is that the majority of the drug targets, they are of course customer driven. They determine those structures that customers want are kinases. Simply a reflection of the fact that the kinases are in the focus of pharma companies. Well, I skip this and end with a second spin-off that is now 12 years old, which has an entirely different business plan, namely the development of a new therapeutic protein. The work again is based on academic work that we did in the 70s and in the 80s on antibodies. We produced the first antibody structures at that time. Antibodies are these Y-shaped molecules. There is antigen binding on the tips, and there is the FC part. Now, what are antibodies doing? They trigger the cellular immune response. So if an antigen binds, if bacterium, virus, any antigen binds to the antibody, then there is a cellular immune response, and that cellular immune response requires binding of FC to FC receptors on cells. This is a bacterium opsonized by antibodies binding to FC receptor. Now this then triggers the immune response, inflammation, for instance. Now our idea was we had determined the structure of this. That was a prominently published result. This is the receptor, this is the stem part. So we thought, how can we modulate this interaction, prevent this interaction, perhaps with a small molecule that we design on the basis of the surface of the receptor or of the FC part. This did not work out. Then we thought we could use the receptor itself as an antagonist. And this actually worked. So the mechanism is as follows. This is the antigen, maybe an autoantigen in the case of autoimmune diseases, opsonized with autoantibodies, and then there is the soluble FC receptor there. And of course, if this binds to the antibody, there is no longer, these binding sites are no longer available for the cellular receptor. Now we did, still in the academic environment, some relevant autoimmune models in experiments with mice. There are mice or rats that develop rheumatoid arthritis, multiple sclerosis, and systemic lupus. Now on that basis then, investors became interested, and preclinical phase 1, phase 2 experiments with some diseases started. Well, another, within that same company, another approach was not to use the soluble receptor, but to use a specific antibody against the receptor that equally well worked. And again, there was structural information that we could provide. This is the FC receptor, this is the FC part that we saw already here, and this is a specific anti-receptor antibody, which, as you can see, binds to a quite different epitope. So this is important for reasons I have no time to discuss. Come to an end. I show my hometown, Munich, in springtime, not only because I love my home city, my hometown, but also because this is the University of Munich, the old central building. And this is where Laue and Röntgen worked 100 years ago, and Laue discovered X-ray diffraction, which started crystallography, chemical crystallography, and biological crystallography. Thank you.
Thank you, Professor Huber, for the wonderful lecture and the wonderful overview of your field of science, which has become really truly used in all disciplines of biology, chemistry, and the interface of those sciences. We have time for a few questions right now. Please, if you have any questions, the floor is open. Если у кого-то есть вопросы, пожалуйста.
Yuri Fomin, MIPT. Well, usually when one thinks about drug, one thinks about substance. But it's quite obvious that the crystal structure of the substance itself can also affect the performance. What do you think? How strong can be the impact of the crystal structure?
Well, there is an influence, but this we can control and measure. For instance, in quite a number of crystals, we find activity. So crystalline protein material shows enzymatic activity. This, many examples of that. But of course we have to be aware of how much crystal packing may influence function. Then it certainly has an influence in all those cases where there are large motions during the catalytic cycle. These cannot happen in the crystal environment. So what we do in this case is we crystallize the same protein in these different states. Let's say an open and a closed, with or without substrate, with or without inhibitor. The other important approach is, so once we suggest an enzymatic mechanism in which certain residues that we see are involved, then we mutate these and check what functional consequences these have. Now we are very much aware of that. It is a real problem, but we can control it and do control it.
Okay, thank you very much.
Why is a proteasome is a cancer target? Because, you know, my simple understanding that proteasome it's a proteins. Okay, these small pieces somehow lead to cancer. But how? Can you say anything about it?
Why is the proteasome a cancer target? Now, it is a cancer target. It's a fact. It was shown experimentally by hundreds, thousands of hundreds or thousands of pharma companies work on it and actually make good business. The mechanism by which the proteasome exerts inhibition, exerts its effect is still not clear. Now there are several ideas one may have in cancer cells. There is increased metabolism. Increased metabolism, increased protein synthesis, increased perhaps damage of proteins which have to be removed. So cancer cells, for that reason, may be more sensitive to proteasome inhibition, but it's really not clear. It's in the middle. It's big business, and we don't know exactly how it works. I think it's not known at all.
I'm sorry, what kind of animal model was used for multiple sclerosis? Was it an animal model?
It was an animal model. I think it is a mouse model, but
yes.
It is a mouse model.
And how far?
But I can't tell you the mouse strain. I'm sorry.
Okay, and how far are these tests from clinical experiments?
Well, they are. So the soluble receptor, this is the main product of this company, has undergone animal experiments, toxicity studies, that is phase 1, and phase 2 have been finished already some time ago for a certain indication. You see, the problem for a small biotech company is to test its drug for a disease where you have a clear endpoint. Now, multiple sclerosis is extremely difficult in that it takes actually years to follow up. Now, they used thrombocytopenia, which has a simple endpoint, namely the thrombocyte count. So this is what they did, made clinical studies of the outcome was not entirely satisfactory, but also not negative, in that sense that the investors wanted to withdraw their capital. They did not, they supported further. Another clinical phase 2 studies with systemic lupus, which is already a difficult disease. This is still ongoing. So it's really an adventure to enter clinical phases with a company that has an excellent idea, but very limited financial resources.
Oh, yeah. So
let me see what comes out.
Multiple sclerosis is still resistive.
Yes.
Thank you.
It's just a terrible disease. I think in general autoimmune diseases are, I mean, cancer we seem to be able to, not only seem to, we can treat satisfactorily with the kinases, also with the proteasome inhibition, but autoimmune diseases there is no cure, you see. So this is something we have to approach to go new ways to.
Dear colleagues, let's thank Professor Huber again, so in the interest of time and staying on schedule. Thank you very much again.
Right. I think I dismount and
Okay.
so close.
While we are connecting the computer, I'll quickly, that's already done, I think. I'll quickly introduce our next speaker. It is my honor and pleasure to introduce Professor Klaus Palme, who is a professor of biology, studying plant biology at University of Freiburg. He obtained his undergraduate education, undergraduate training at University of Freiburg, and then obtained his graduate degree, his PhD, at University of Ulm, and Habilitation at University of Cologne. After that, he's been appointed to, and held several different positions and had many different opportunities, but ultimately returned to study plant biology and small molecule interaction with the plant proteins at his home university at the Albert Ludwigs Universität Freiburg. And where he currently is also the director of the Center for Systems Biology.
I think we duplicate.
His interests include high throughput screening and interaction of small molecules and small molecule hormones such as plant auxins and auxine, with plant proteins controlling the growth, differentiation, and regulation of plant functions. So, my pleasure.
So thank you very much, and many thanks, for the invitation to be at this really stimulating place. We all enjoy it very much. Now, I would like to take you now on a short tour away from the angstrom level that we've experienced in the last 2 days, up to the nanoscale, and then further to micrometers, millimeters, to organs, so to speak, and back to cells. Because, of course, all the proteins that we've seen so nicely at this high resolution do, of course, need the cellular environment in order to work. And this is, in fact, where we are interested in. How do the proteins work, and how can we interfere with this in order to understand the mechanisms of certain processes that allow, in fact, the cells to develop and to adapt to the environment. And this is, in fact, just a very brief introduction into the organism that we're working on. And of course, I mean, you can do everything also with animals, but many aspects of methods development are much easier in plants, I have to tell you. I've been working for quite a number of years with animals and animal viruses. So in other words, using plants has quite a number of advantages, and one of them is, of course, I mean, they do not need a very complex environment to grow. And of course, the plants are sessile, they are stand somewhere, and they have to cope, in fact, with all the environmental challenges. That can be the light, too much light, too less light. It can be the temperature being up and down, the air quality, the water which may be lacking, or where there may be too much water, the soil conditions, physical barriers, all of that. And this gives, in fact, I mean, the plant not only challenges, but also opportunities to act, to react, in order to be productive, to deliver seeds, or, I mean, to be used very efficiently by the farmer in order to produce our food. So this is, in fact, I mean, the situation. And in order to do this, the plants have very complex signaling pathways. And they're not only having complex signaling pathways with many gene families that we know to be important in humans being expanded by a factor of 10 or more. We learned yesterday that human cells have about 500 kinases. Of course, plants do need more. They have about 1,500 to 2,000 kinases. They have many other protein families which are extended, and this illustrates, in fact, the specificity of control mechanisms. So many metabolites being synthesized, plants being chemical engineers, producing numerous chemicals, thousands of chemicals in order to regulate their pathways or to interfere with other organisms in order to fight them. And therefore, in fact, I mean, plant signaling is extremely complex. You see that here with our model organism, a very small plant, which actually is a wheat. Just going back. And this wheat can be found outside. You have all the features of a typical plant, but many mutants allow you, I mean, to modify the plant in order to study the mechanisms. So I don't want to give you a plant lecture, but just to illustrate the scales that we have, I mean, from the nanoscale through the micrometers up to meters, actually. And this you see here, where we have a typical root on the right side, and on the left side, again, our wheat, which gives unique opportunities to search for new mechanisms in order to control growth. So the challenge being to describe, in fact, I mean, the cells and the interior of the cells in the context of cell layers and organs, basically in 3 or 4 dimensions. If you take the time as another dimension. And this brings us back, in fact, to a situation in microscopy where we need, of course, methods to look at very high resolution into cells. And within the past few years, there have been enormous progress. Now, the challenge, in fact, was that to a certain extent, I mean, microscopy about 10 years ago was, in fact, like the development of cars in the last century. So this gives us, in fact, the situation where about 100 years ago, the German father of Mercedes, he was just saying that the worldwide quest for cars will not be more than 1 million, because there will not be enough drivers. Now, coming here to the institute this morning, we already learned, that he was absolutely wrong. Yeah. So, in fact, it is just a way, to simplify the machines, yeah, to make them, in fact, robotic, yeah, in order to make them useful for everybody. And this is, in fact, exactly the situation that we have at the moment in the microscopy. Namely, today, again, 1.6 billion cars, yeah, the chauffeurs, yeah, we can even get rid of the chauffeurs by completely automating the process, yeah, and with the Google car, you just sit in and and the car will drive you without any driver. This is actually what we would like to have in microscopy, where in fact the microscope I could just here from the stage in Freiburg control and just look into the processes there. Yeah? So will that be possible? Yeah? Well, this is how a microscope looked about 150 years ago. A very simple instrument. Yeah? And then over the past, say, 30 to 40 years, microscopes got more complex. They had many features, and you can buy these complex instruments from many companies, be it Nikon, Olympus, or some German companies like Zeiss. Now, again, coming back to the robotization, namely to go through a high throughput to use them in order to produce images like spectra in chemistry, which are complex in terms of information, just to read this out. Now, for that, you need, of course, or you would like to wish, in fact, a simpler instrument, which would be much more robust. So, in fact, expert instruments. But at least, the commercial ones are not so easy to be integrated in completely automated workflows. Yeah? So what is needed is something which is extremely simple, which should be plain, and it should be able to be implemented into robots. And this is, in fact, what you see in this black box. Yeah? And the inventor and the collaborator of us is Rainer Uhl, with whom, in fact, the 3D capabilities of a confocal microscope were introduced into this small box there, just making it, compared to other microscopes, much faster, giving it more life, namely less photo damage in the tissues, integrate, in fact, this into an automated workflow, and basically introduced adaptive intelligence, computer intelligence, in order to control it. So here you see the output of this, where in fact the German Space Agency decided to put this instrument even on a rocket, which will fly soon. And this microscope, it's a spinning disk confocal microscope, does even withstand all the stresses that occur during these rocket starts. So it has already been successfully flying in parabolic flights and soon be used for live observation of cellular reactions under space conditions. So what is inside in this small box? You see here a lot of small parts where in fact there are the light sources, LEDs, lasers. On the right side, you see the unit in which the imaging occurs. And this can be used for confocal analysis. You can even, go further by using structured illumination, where you rotate grids along an axis, where through the interpretation of the patterns that are caused by these rotations, basically interferometric responses, you can increase the resolution up to a level of about 100 nanometers. And by using this, one has basically an 8-fold better resolution in 3D than with a standard microscope. And you can even improve it, and that has been introduced here, to super resolution, where in about 80 microseconds, you can generate about 50 planes at a resolution of about 120 nanometers. That's breaking, in fact, the diffraction limit of light, which is in the range of 250 nanometers in the Z direction. Yeah? So controllable like a car, indeed, yes. You just take a mouse and an iPad, and with this you can easily control this machine even from distances like here from Moscow to Germany. So there's no problem. You can directly look to the images and interpret them. So in other words, you can take an educated computer and use machine intelligence in order to run completely automated robotic stations where you can use chemical genetics, where you can implement various technological features where the robot puts in the plates with your cells to be analyzed for generation of huge numbers of images, where in fact overnight, you create a terabyte or more of data just from a single microtiter plate, each image being a spectrum of a cell, which you then need to interpret. And now, in fact, there comes the point for the computational scientists, the computer scientists, to interpret these images in order to dissect them into their elements and use these elements backwards to build up the individual frames. So what we are now just looking. So the machine opens, the robot inserts the plate, and then the pictures are taken automatically in 3D. So that's, in fact, what it does. So there's a little bit of a There's a problem here with the... Just going to get out of control. So I will now give you a couple of examples of this journey. So here we're looking, for example, on the top of cells analyzed by structured illumination at a level of resolution in the range of about 100 nanometers. So you see here all sorts of structures on the surface of the cell, which can, of course, be interpreted in biological means, but this is not, the topic of today's talk. So, the resolution is enormous. The information is enormous, and you can use this for interpretation. Here you have, for example, the analysis of a cell cycle over time, where the different phases up to the cell division, where it freezes over there on the right side, then cell plates are introduced and the cells are dividing. We can follow the cells with many different reporters. These give these nice colors, indicating either members of signaling pathways or cellular structures. But then, of course, what to do with this? I think the interesting point is that we now can quantify cellular information. Basically, what we obtain is a quantitative description of what happens in cellular cohorts. Basically, we learn how many cells are growing and dying, what is happening with respect to size and shape, and what, in fact, the form changes during cell divisions are. And this allows us to introduce, to combine shape and space by optical flow methods. So this gives, in fact, a little bit of indication that there's a lot of mathematics in the back ongoing in order to describe these processes there, where you then can follow, in fact, the changes in the individual cells in cohorts, and you get the statistical behavior of these cells in relation to specific pathways as they are ongoing. But then further, what can one do with this? Basically, you can build a synthetic cell. All the informations, all these spectra, which images are, can then be introduced back by using machine learning methods in order to extract cell behavior from images. So in other words, I mean, the images or movies can be used and in order to train, in fact, the properties of a cell, to integrate all the cells, all the elements in order to get, in fact, a statistical interpretation of the different processes that are happening in the cells. And this is the work of one of our collaborators, Bob Murphy, and his team. This can be used in animal cells, likewise in plant cells. And you see here, in fact, how PC12 cells undergo, in fact, differentiation over time. These are, in fact, reflections of transcriptome patterns. And these changes in form can then be related down back into individual reactions within the cells, and thereby we capture important information. At the same time, we can use these technologies to improve screenings. What you see here in the bottom line is, in fact, a chemical screen done in the random fashion as it does nowadays. So basically, I mean, today's chemical screens are rather inefficient. But by using machine learning methods in order to interpret the cellular features that are ongoing, you're getting better and better. So these are the green lines, and by using active learning procedures, basically within a much smaller number of individual screens, you get much better efficiencies in terms of steps that you have to do, that you have to execute in order to extract the most valuable compounds with respect to the cellular phenotype you want to search. So back from cells to organs. So here you see a root, and there, in fact, we want to come down to a cell and then come backwards from the cell again to the organ. So in other words, here you can do, in fact, protein quantifications. You can determine the protein interactors or modifiers, and bring them back, in fact, to growth and patterning. So how to do this? Basically, use again imaging in order to look through a tissue and then extract all the tissue properties, all the cellular properties. You determine, in fact, where the nuclei are. These are here, these blue candidates, are extracted here in this cartoon, where you have the different cells. And you can then use, in fact, the data that you extract from the images in order to build a three-dimensional structure of a whole tissue or organ. So in other words, I mean, what you want, to do is to work inside of such a tissue. These are just the nuclei shown by in this cartoon for clarity. You are observing, in fact, each of the nuclei, each of the cells in their full structure. So what you get from here is the position of a cell within the cell layer, each of the informations, like dimensions, length, width, and thickness, where the nucleus is with respect to GPS coordinates, the mitotic status. All these informations can be extracted here from the images. And by this, you introduce all of this into your computer programs that you develop, and you're able, now to follow, in fact, the organ as you can see it from outside by regular imaging, now in the 3D structure is composed from the imaging. So this is, in fact, the reconstructed organ. And we're now just moving, in fact, and turning around the organ as we like. And we now go micrometer by micrometer. Inside, we take just a few cells away. We now look into the root. Here we have the so-called pericycle cells. This is the cortex. Now we take the look to the so-called endodermis. So cell by cell, you can now follow, in fact, the information in structural way on how, in fact, the different cells make up the organ. And basically, such information is important to understand on a phenomenological basis how certain compounds, how certain pathways are affecting the growth, development, and differentiation. Of course, the same thing can be done with animal organs as well. Just having a few looks inside. So this is information that gives you a very high resolution but also high content opportunity to analyze cellular processes. It allows you to look to organs, to cells at different dimensions and scales, ranging in fact from nanometer to micrometers. And the computational analysis and the algorithms developed improve the efficiency of the chemical screens enormously. So this is in fact the message I wanted to bring over. Thanks to the teams, mostly computer scientists involved, but also biologists in Freiburg as well as internationally, and our money providers are basically the European Union, the European Space Agency, and other companies. So thank you very much for your attention.
Thank you indeed, Professor Palme, for the very stimulating, wonderful lecture. We have time for a couple of questions, if there are any, from the audience. I'll just say it as a comment. It's the technology, the imaging technologies of today are amazing, of course, and that's mostly the technological breakthroughs. But what you can see in terms of the biological processes and chemical processes that are happening in cells in real time, as the plant is developing or as a specific tissue is developing, it really puts the science at a different level. What we can observe in dynamics and as a material is growing and being produced. It really is breathtaking.
How do you come around avoiding photo damage? Because you said you do not damage tissue by doing all that.
That's a very good question. Normal confocal microscopes are illuminating the whole sample. So that means that through the whole depth of the tissue, you're putting a huge amount of laser light. With the structural illumination, you're just going in certain frames, and you're not illuminating the back and foreground. And that reduces, of course, the photo damage dramatically. Or you use, in fact, light sheets, where you're again, reducing the photo damage due to not illuminating the whole tissue. Now, in this case, it depends on the particular setup. So I've been introducing here a couple of different setups. Where, if you go with a standard confocal microscope, you have, of course, the difficulties, you're mentioning, If you go for light sheets, for example, you're reducing this. Likewise, for the structured illumination, Okay.
Right. If there are no further questions, please thank again Professor Palme for a very stimulating lecture. Discovery has many different phases, and we are all familiar with the purely scientific discovery in this audience and looking at the biological aspects of the living organisms and plants and putting together our understanding of the conceptual and the very detailed level of how molecules work and how tissues form and so forth. But there is a raw discovery as well, so the discovery akin to what Magellan and Cook did many years ago and what the last frontiers or the frontiers which are left for us are the, for example, the exploration of the space. And we have a very special guest. We have a very special treat today. A very famous Russian cosmonaut, Sergei Krikalev, who is going to address you and who is going to say a few words to the students, to the audience, about his thoughts and reflections on the science, because like I said, the discovery, scientific, technological, has many different faces. Sergei was educated in Leningrad and now St. Petersburg at the Leningrad Institute of Mechanics, and after that worked at the very well-known organization for integration of science and technology, Energia, and after that point was selected and was trained to become a cosmonaut. And on the very first mission, he went in 1988 on Soyuz 7 mission, which lasted 151 days, I believe. And then one of the longer ones in 1991-92, which is a very interesting, has very interesting history. Read it, I encourage you, you'll enjoy it. He stayed in the space for 310 or 311 days, I think. And then was one of the very, not one, he was the first Russian cosmonaut to go on the STS-60, the Discovery shuttle mission, with US NASA agency. And this is just a fraction of the record, but I can tell you that he holds the absolute record of being in space on the space station for over 803 days. He had 8 outer space missions. His accomplishments have been recognized by many different awards, including the Hero of the Soviet Union and the Hero of Russian Federation. Very select, very few people hold both honors, both titles. So it really is a distinct pleasure and an honor for me to welcome you to the stage. Thank you. If you don't have a translating device, the lecture, the presentation is in Russian, please raise your hand and pick up the translation device.
Спасибо за представление. Уважаемые участники конференции, мне очень приятно здесь быть, я рад вас приветствовать, и очень приятно присутствовать на такой интересной конференции. Я вижу, что здесь много науки интегрируются вместе, и, наверное, в дальнейшем из каких-то отдельных частей, особенно на стыке, может быть, различных наук, будут появляться новые технологии и новые прорывы в движении вперед. Как здесь уже было сказано, космонавтика интегрирует в себя очень много различных дисциплин, и где-то мы опираемся хорошо на известные факты, где-то мы вынуждены идти вперед, может быть, ощупью, и продвигаться вперед, продвигая человечество за пределы существующего уже ареала обитания. И так было всегда. Люди выходили из пещер, может быть, не совсем зная, что находится за пределами пещер, исследовали и продвигались вперед. Люди переплывали реки, переплывали океаны, обнаруживали другие земли. В конце концов, совсем недавно поднялись в воздух и получили другую возможность уже передвигаться. И вот настал черед следующего шага вперед, шага за пределы земной атмосферы, шага с поверхности Земли за пределы нашего привычного ареала обитания. И, может быть, к сожалению, конечно, мы все-таки сейчас пока в таких небольших капсулах летаем за пределы атмосферы, уже достаточно долго существуем за пределами земной атмосферы. И, конечно, вот это движение было невозможно без движения науки, без тех знаний, которые мы получили для создания космических кораблей. И после того, как мы научились запускать ракеты, как мы научились выходить за пределы атмосферы, довольно быстро мы послали туда человека. Сейчас это кажется небольшим, может быть, шагом вперед, когда всего один виток, всего 108 минут длился первый полет. И по тем временам, конечно, было много неизвестного, потому что сейчас, кажется, это все легко и просто, но если обратиться к тому, как принималось решение о посылке первого человека, какие первые эксперименты ставились в космосе. Первыми экспериментами было, может ли человек глотать в космосе, может ли человек нормально дышать в космосе. Сейчас это кажется немножко наивным, но это показывает ту степень неопределенности, в которую двигались первые космонавты. Конечно, потом и космическая биология, космическая медицина дали немножко больше знаний. Мы научились летать, и не просто летать, а длительно летать в космосе. И для длительных полетов проводились большое количество исследований, которые позволили делать эти шаги все более и более длительными. Как здесь было сказано уже, мы говорим про рекорды длительности пребывания в одном полете, рекорды суммарного пребывания. И все это еще, конечно, мы до конца не знаем. Мы еще не знаем окончательного влияния космоса на организм человека. И вот делая такие постепенные шаги, мы потихонечку продвигаем вот эту границу нашего знания и границу возможного обитания. Кроме того, что наука и космическая биология, космическая медицина дают нам некие знания и уверенность для того, чтобы мы двигались вперед, сами условия космического полета вообще являются уникальными. Здесь говорилось о тех экспериментах, которые ставятся на Земле, о том, как современные технологии позволяют нам заглянуть внутрь клетки или получить детальные знания об отдельных белках. Такие же эксперименты проводятся в космосе. В космосе уже на протяжении многих лет, начиная со станции «Салют», очень широко это было представлено на станции «Мир», и сейчас подобные эксперименты проводятся на Международной космической станции. Идут исследования по выращиванию кристаллов, белковых кристаллов, которые позволяют исследовать их уже немножко на другом уровне, на макроуровне, не влезая в ангстремные размерности. Проводятся эксперименты как над отдельными белками, которые, может быть, в будущем позволят получать лекарства, так и на технологиями, потому что условия космического полета, условия невесомости позволяют получать более крупные белковые кристаллы, позволяют получать новые технологии для получения сверхчистых веществ, для получения сверхчистых лекарств. И вот эти технологии сейчас и отрабатываются в космосе. И, конечно, новые знания нам нужны для того, чтобы двигаться дальше. Сейчас уже активно обсуждаются полеты за пределы земной орбиты. Такие пробы были сделаны в конце 60-х, начале 70-х годов, когда американские астронавты летали к Луне и высаживались на Луну. Но сейчас и в Соединенных Штатах, и в России в космических агентствах обсуждаются будущие программы, когда человечество полетит за пределы земной орбиты надолго, когда мы, видимо, будем создавать, и мы, американцы, и сейчас уже и китайцы планируют создание напланетных космических станций, таких как база на Луне, может быть, когда-то в следующем на Марсе. Но для того, чтобы нам долететь до Марса, нам потребуется еще достаточно много исследований. Эти исследования должны быть для понимания того, как человеческий организм сможет преодолеть те трудности и те проблемы, которые нас ограничивают при полете к Марсу, такие как повышенная радиация, такие как стесненные условия космического полета. Поэтому какие-то эксперименты, такие как вот Марс-500, например, проводятся для того, чтобы понять, как человек психологически сможет перенести такой полет на Марс. Потребуются, видимо, дополнительные исследования и космического пространства, и возможности человека, и создание систем защиты для того, чтобы понять, как человек сможет долететь до Марса и при этом не получить смертельно опасную дозу радиации. Таких исследований еще предстоит много, и я думаю, конференции, вот типа сегодняшней конференции, позволят по крупинкам собирать эти знания, которые позволят нам двигаться дальше вперед. Еще раз рад вас здесь приветствовать и спасибо вам за внимание.
Спасибо, Сергей Константинович. Если аудитория, используйте возможность задать вопросы. Мы вернемся.
Если есть вопросы.
Молодое поколение, пожалуйста.
Скажите, пожалуйста, вот одно из сильнейших изменений, связанных с космосом, кажется, связано с мозжечком. Можно ли с вами будет просто детально побеседовать?
Ну, наверное, я здесь все-таки здесь лучше, наверное, пообщаться, если настолько детально, все-таки с Институтом медико-биологических проблем.
Ваши личные, вот интересно, ваше личное впечатление. Ну, вы представляете, наверное, что такое?
Ну, да. Ну, да, может быть, тогда более специфичный вопрос,
пожалуйста. Да, тогда просто, после этого попытаться с вами поговорить. Спасибо.
Сергей Константинович, вопрос. В прошлый раз вот не дошло до меня задать вам такой вопрос. Ваш первый выход в космос. Ваши мысли перед тем, как открыть дверь, и ваши мысли после того, как вы ее за собой закрыли, вернувшись на корабль.
Ну, я хочу сказать, что как и космический полет в целом, особенно, конечно, первый полет, первый выход в космос — это очень такое впечатляющее событие, я бы сказал. Здесь мне трудно, наверное, приводить какие-то аналогии, хотя, ну, может быть, аналогия, знаете, какая? Как так получилось, что когда я первый раз выходил в космос, я уже к этому времени достаточно пробыл в космосе. Мы на первой экспедиции готовились к серии экспериментов за пределами космической станции. Мы готовились к испытанию нового маневрирующего, как это тогда называлось, кресла, хотя это кресло скорее похоже на шкаф, который цепляется сзади на скафандр. Там стояла реактивная система управления, автоматика некоторая, которая позволяла космонавту, как вот на скутере люди перемещаются по воде, вот точно так же вот на этой космической ранцевой установке перемещаться за пределами станции. К сожалению, было проведено много тренировок, но программа полета изменилась, и так бывает, к сожалению, в полетах. Программа изменилась, и мне не пришлось выходить в моем первом полете, поэтому, когда я первый раз выходил в космос, это уже был мой второй полет, я уже имел достаточно опыта полета за плечами, и тем не менее, вот выход в космос — это все-таки очень впечатляющее событие, потому что аналогия здесь может быть такая: когда мы летаем рейсовым самолетом и в иллюминатор смотрим Землю, вроде кажется уже все понятно, и это знакомый вид, но если кому-то приходилось прыгать с парашютом, или, может быть, у людей достаточно фантазии, чтобы это представить, но когда ты подходишь к открытой двери у самолета с парашютом и готов прыгнуть туда, вид Земли, я вам скажу, видится совсем по-другому. И ощущения совсем другие. И поэтому, и, кстати, в чем-то, наверное, схоже, потому что я помню свой первый парашютный прыжок, когда я с парашютом вот подошел к этой двери перед выходом за пределы самолета, и я понимал, что я, конечно, не первый и не последний, кто прыгает с парашютом. И с таким же или с этим же парашютом прыгали уже другие люди. Система достаточно надежная. Но все-таки приходится делать, несмотря на то, что я психологически к этому был готов, приходится делать достаточно много усилий для того, чтобы таких психологических усилий, чтобы шагнуть за пределы самолета, потому что все наше существо, вся предыдущая история человека как вида на самом деле этому противоречит, чтобы вот так вот спрыгнуть с большой высоты куда-то непонятно куда. Примерно то же самое происходит и когда выходишь за пределы космического аппарата, ты уже привык к виду Земли через иллюминатор, но когда открываешь люк и выходишь туда, это совсем другие ощущения. Вот сродни чем-то с ощущением с первым прыжком с парашютом. И я помню, что когда достаточно хорошо застрахованный, когда двумя мощными карабинами, мощными фалами ты прикреплен к поручням, когда ты осторожно перемещаешься наружу, перецепляешь сначала один карабин, потом второй, понимаешь, что каждый из этих карабинов имеет несколько, или каждый фал имеет несколько тонн на разрыв, тем не менее руки все равно отпускать не хочется. И вот первые требуются какие-то, может быть, несколько минут или секунд для того, чтобы адаптироваться, чтобы начинать дальше перемещаться. И, конечно, руки тебе нужны не для того, чтобы держаться за поручни, а для того, чтобы выполнять ту работу, которую нужно делать. И эти работы были интересны. Они относились не совсем к теме этой конференции. Мы строили большую конструкцию. Собственно, это был один из таких интересных экспериментов, как построить большую ферму. Технологий существовало много, делали это американцы. Они собрали одно из звеньев такой большой фермы, поняли, что это очень трудоемкая операция, но получили как бы отрицательный результат этого эксперимента. Наш эксперимент был более положительным. Мы собрали 14 звеньев фермы. По тем временам это была тоже рекордная длительность конструкции, собранной в космосе. Мы использовали тогда очень интересную технологию сборки, используя материалы с памятью формы, которые, собственно, позволяли делать эту конструкцию. И, собственно, экспериментом являлось первое, можно ли ее собрать. Второе, если мы соберем, будет ли она достаточно жесткая, потому что потом мы снимали характеристики вот, собственных колебаний, жесткости этой конструкции. И если она окажется жесткой, на нее предполагалось установить, и так потом и случилось, дополнительную двигательную установку, которая в разы уменьшила расходы топлива по крену. То есть это было одной из тоже таких технологий продвижения человека за пределы нашего ареала обитания, чтобы научиться строить конструкции в космосе, более эффективно летать и по возможности летать дальше. Ну и когда вернулись, на самом деле это довольно трудоемкая работа. Вообще, ну, может быть, тогда чуть больше о том, что такое работа в скафандре, потому что скафандр — это маленький космический корабль, и внутри этого корабля мы вынуждены держать пониженное давление, там где-то 0,4 атмосферы. Это вынужденная мера, потому что если бы мы держали давление больше, то этот скафандр было бы невозможно свернуть. Вы представляете себе, что если вот рукав — это как надутый футбольный мяч, его надо сгибать каждый раз, это довольно приличные усилия. В конструкции скафандра там есть специальные меры, принимаются для того, чтобы все-таки его можно было согнуть, чтобы можно было согнуть пальцы, но после 5-6 часов работы в скафандре, когда ты каждое твое движение ты должен сгибать скафандр, сминать фактически вот эту оболочку, это довольно трудоемкий процесс, и возвращаешься с этого выхода, ну, физически измотанный очень сильно заряжены эмоционально, но тем не менее, несмотря на то, что это физически очень сложная работа, каждый из космонавтов всегда хочет выйти наружу, потому что это очень такая эмоциональная работа, это другой опыт, который мы получаем в процессе космического полета. Ну и возвращаясь назад, конечно, это вот ощущение сделанной работы, завершенной. В общем-то, выход в космос — это и дополнительный риск, потому что, наверное, риск чем-то соизмерим с альпинистом, который лезет по вертикальной стене. Да, он лезет, страхуется, но в случае ошибки он может отделиться от стены, упасть в пропасть. В нашем случае также ошибка вот в страховке себя или оборудования может привести к тому, что ты отделишься от станции. Но в отличие от падения альпиниста, которое занимает там несколько секунд, это свободный полет, падение будет продолжаться несколько часов, но ресурс скафандра не такой большой, это реальная как бы угроза человеческой жизни. Поэтому работа проходит в таких в реальных, физически тяжелых и, в общем-то, в какой-то мере стрессовых условиях. Ну и, как всегда, после стресса, конечно, какое-то чувство облегчения, что ты завершил свою часть работы. Прошу прощения за длинный ответ. Еще какие-то будут?
Можно? Извините, мы тут чуть-чуть поспорили, конечно, я не разделяю мнение профессора Хубера, да? Фундаментальный вопрос. Технология сейчас позволяет на самом деле все делать роботами. Вот зачем туда отправлять столько людей и рисковать людскими жизнями?
Вы знаете,
ну, роботами же все можно, это все эти операции, вот растения туда-сюда, да, как вот биологический эксперимент. Это же все можно сделать, в принципе, сидя на земле. Вот это чисто вот наше любопытство, вот выйти, не выйти, или все-таки...
На самом деле хороший вопрос. И, честно говоря, я с самого начала, как бы занимаясь именно пилотируемой космонавтикой, сам себе задал вопрос, типа, а зачем мы летаем? Потому что это вопрос скорее философский и вопрос такой стратегический. Я совершенно с вами согласен. Вот эта идея работать за пределами или в опасных условиях роботами или человеком, она вообще на самом деле достаточно старый вот этот спор. И точно так же, когда американцы высаживали человека на Луну, а мы туда запускали луноход, но всегда были сторонники автоматизированного или роботизированного подхода и человеческого подхода. И, наверное, истина лежит где-то посередине. Вот если говорить об эффективности, да, уже не просто об одном конкретном, там, одну конкретную операцию действительно может быть проще выполнить роботом. Но чем более сложной становится задача, тем вот этот оптимум сдвигается более в человеческую зону. Потому что когда задача становится настолько сложной и становится очень сложно запрограммировать робота, мало того, очень сложно обеспечить надежность этого робота, то выполнение сложных задач оказывается проще решать уже в некой комбинации человека и машины. Кстати, может быть, хорошим примером является работа американского телескопа Хаббл. Он с самого начала делался, с одной стороны, это автоматический аппарат, и очень часто мы слышим критику от ученых, что все-таки станция — это не самое хорошее место для размещения высокоточного оборудования. К примеру, вот такие примеры, как Хаббл, который может позволить себе навестись и, может быть, там, минутами или даже часами смотреть в одну точку неба, набирая свет с этой точки неба, на пилотируемой станции делать не очень удобно, потому что все-таки станция делается и для человека тоже. Есть некие вибрации, есть ограничения на развороты этой станции. Существует, будучи таким сложным объектом, станция подчиняется другим задачам и другим критериям, поэтому не всегда и не все эксперименты удобно проводить на станции. Но тем не менее, я могу привести пример попытки роботизировать одно из исследований, в частности, исследование неба в ультрафиолетовом диапазоне, которое мы проводили, вот как здесь было упомянуто, в одном из первых полетов, в первом моем полете, в 1988 году еще на станции «Мир», когда был размещен ультрафиолетовый телескоп, и была некая оценка, какими экспозициями мы должны получить изображение неба в ультрафиолетовом диапазоне. Когда получили первые пленки с автоматического режима этого телескопа, то информации там было 0. Да, и если говорить про эффективность, некую меру эффективности, да, отношение результата к затратам, то затрат, в общем-то, для автоматических аппаратов мы говорим меньше, чем для тех, которые работают на пилотируемых комплексах. Этих затрат меньше, поэтому знаменатель у нас меньше. Но когда в числителе 0, то вот эти затраты становятся уже не столь важными. То есть получили нулевые данные. И слава богу, этот аппарат позволял работать в полуавтоматическом режиме. И фактически методом ощупью начали подбирать эти экспозиции, и в конце концов нащупали те экспозиции, которые дают значимый результат. И потом он продолжил дальше работу в таком полуавтоматическом или в автоматическом режиме, но вот этот поиск осуществлялся вручную. Для примера, тот же самый Хаббл, будучи выведенным в космос, оказалось, что недостаточно сфокусированы. Что-то не учли на Земле, это нормально, но огромный, дорогостоящий, сложный аппарат, у которого, кстати, в знаменателе вот эти затраты были очень тоже большие, его результат был близок к нулю. И только то, что с самого начала закладывалась возможность ремонта пригодности этого аппарата, то есть некая комбинация человека и машины, позволила, во-первых, доработать его для того, чтобы получать высококачественные данные. И, кстати, много лет этот аппарат работал, потому что была возможность менять аппаратуру. С самого начала были какие-то каналы управления этим телескопом резервированы, но конструкция аппарата, будучи резервированной, все равно оказалась недостаточно надежным для того, чтобы обеспечить сбор этих данных на протяжении многих лет. Поэтому несколько миссий к этому телескопу, которые, в общем-то, тоже были достаточно дорогостоящими, они позволили продлить длительность этого уникального аппарата и получить очень высокозначимые научные результаты. Поэтому я думаю, что дальнейшее движение — это будет некий компромисс или некий симбиоз человека и машины. Какие-то задачи будут решать машины, какие-то задачи будет решать человек. И чем более сложные задачи, чем менее определенные проблемы будут у нас стоять перед нами, тем больше влияния будет иметь человек. Поэтому я думаю, вот эта вот комбинация. Машины хорошо решают хорошо детерминированные задачи. И чем более сложные области мы уходим, тем большую роль будет играть человек. Поэтому я думаю, в будущем вот такая комбинация человека и машины окажется наиболее эффективной.
Спасибо.
Да.
Сергей Константинович, скажите, пожалуйста, вы достаточно длительный срок проводили в космосе. По возвращению на Землю, как долго проходила ваша реабилитация?
Длительные полеты мы считаем, все-таки есть у нас некая такая тоже градация. Короткие полеты, мы уже не говорим даже про полеты, там один виток. Считается, что полеты меньше 1 месяца, мы, такая грубая классификация, считаем короткими полетами. Полеты более 1 месяца считаются длительными. Вот, как я уже говорил, мой первый полет был около 5 месяцев. Действительно, организм значительно меняется в космосе. Меняется состав крови, меняется количество крови. Кстати, я не знаю, насколько это известный факт, мы теряем за длительный полет, вот такой около 5-месячного, порядка 20% крови в процессе полета. Начинает вымываться кальций из костей, меняется мышцы, структура мышц. И все это потом требует определенного восстановления. И, честно говоря, вот я вспоминаю свои ощущения, особенно после первого полета, когда все это встречаешь первый раз, когда всю жизнь себя привык ощущать таким сильным, здоровым, способным спрыгнуть с пару метров высоты, 3 метров высоты, перекатиться, встать и пойти дальше. И вот когда ты после первого полета, я помню вот это ощущение, что я понимаю, что спрыгнув со стула, я получу реально травму, вот это несколько такое необычное ощущение, и действительно это требует какого-то времени для такой физической реабилитации. У нас вот этот процесс разбит на два этапа. У нас есть некий, то, что мы называем острым периодом реабилитации. Это порядка 3 недель, когда мы должны вот после полета иметь возможность ходить, стоять, двигаться так, как вот нормальный обычный человек. Потому что поначалу, вот вы видели, наверное, съемки после полета, когда космонавтов там фактически на руках вынимают из аппарата, и очень часто мне задают вопрос, а что действительно вот люди не могут встать и ходить? Могут. Но ощущение вот примерно такое, как при двукратной перегрузке. То есть вот отвыкнув за несколько месяцев от полета, приходится напрягаться, но можно встать, можно идти. И проблемы, кстати, основная проблема не столько в крупной мускулатуре, которая двигает нашим скелетом, сколько в мелкой мускулатуре, которая управляется вегетативной системой, которая держит тонус сосудов, которая позволяет выкачивать кровь наверх. И крупные мышцы мы можем тренировать в космосе, а вот эти мелкие, на самом деле, труднее тренировать, и их надо восстанавливать, потому что когда человек встает, он может встать, но остатки крови опускаются вниз, и их нечем выкачать наверх, поэтому действительно такой процесс. Встал, чуть-чуть походил, полежал, снова встал, и вот эти вот периоды хождения становятся все более длинными, периоды лежания все более короткими, и вот по завершении 3 недель мы как бы уже способны нормально ходить, как нормальные люди, без ограничений. Но тем не менее бегать, прыгать еще все равно возникают проблемы, и это как бы второй период реадаптации, который занимает около 40 суток, 30-40 суток, когда мы по окончании этого этапа уже можем бегать, прыгать, как нормальный средний человек. Может быть, не так, как это было, когда ты был до полета, но уже вернуться на нормальный уровень, с которого ты можешь дальше восстанавливать уже свою физическую форму. И, честно говоря, отдаленных последствий мы еще до конца до сих пор не знаем, потому что, даже по собственным ощущениям, что вроде уже адаптировавшись, начинаешь заниматься каким-то спортом и чувствуешь, вот оно какое-то есть ограничение, там уже прыгать в волейбол, так как я прыгал до первого полета, через там 3-4 месяца еще вызывает проблемы. Как говорят медики, и по тем экспериментам, которые они проводят, какие-то изменения в костях, они понятные. Вымывание кальция — это легко зафиксировать, это вымывание и восстановление этого кальция, но структуру этого восстановления на самом деле мы до конца еще до сих пор не знаем. Да, проводятся какие-то эксперименты на мышах, когда с помощью гипокинезии у них вызывают те же самые проблемы с вымыванием кальция из костей и потом восстанавливают. Но эти опыты показывают, что структура костей становится немножко другая. Я, имея сейчас рекорд длительности полета, пока еще не готов представить свои кости для детального анализа. Поэтому я думаю, науке придется еще немножечко подождать.
Спасибо.
Сергей Константинович, мы знаем, что вы недавно вошли в Русское техническое общество, да, возглавили экспертный совет. А планируете ли вы как-то сотрудничать с Физтехом?
Да,
может
и
быть,
это одна из
систем по другим направлениям.
тем разговора. На самом деле, действительно, вот из тех разговоров, которые мы сейчас ведем, становится понятно, что очень много интересных результатов можно ожидать как раз на стыке наук. И, может быть, одной из востребованных вещей, которые есть, это возможность дать специалистам разных областей общаться между собой. Понятно, что уже сейчас видны какие-то проекты, которые могут подвинуть вперед, может быть, какие-то вещи, связанные с технологиями, какие-то вещи, связанные с созданием более комфортной среды обитания, более совершенного транспорта, но одна из задач, которая есть, это вот коммуникативная площадка, где ученые и специалисты одной области смогут общаться между собой, но и также общаться между разными областями. Поэтому как раз сейчас идет обсуждение этого процесса, как это будет выстроено, покажет время, но вполне очевидно, что такая